Hi guys, I am Cassandra and we'll be doing experiment 19 today, electrochemical cells. So part A of the lab, you will be watching a video posted on Blackboard, and this is the electrochemical reaction of potassium iodine and phenolphthalein. Make sure that you observe the video and record your observations. And today you will be doing several electrochemical cells. You're going to be measuring their potential with a voltmeter. The calculations and equilibrium constants and free energy changes will be measured using cell potentials. These cells will be under non-standard conditions, and that means that they're not going to be at 1 molar and the temperature will not be 25 degrees Celsius. And usually electrochemical cells will be connected to complete a circuit. The reduction occurs in the cathode and the oxidation reaction will occur in the anode. To start, you will make sure that you have your safety attire. You will need goggles, gloves, and the lab coat. And anything below the waist should be completely covered. And you should be wearing long pants and toes covered. The materials that we will be needing today, we'll be needing copper sulfate solution, zinc sulfate solution, and iron sulfate solution. You will be needing nitric acid as the cleaning solution, several beakers. You'll be needing the porous cup, the electrode strips, and thermometer. You will also be needing tongues. To begin our experiment, you will need to grab the porous cup and dip it into the nitric acid solution. All right. Make sure it is fully immersed and rinse it as best you can. And then we're gonna rinse it with DI water as much as we can. After cleaning the electrodes and cleaning the porous cup, you will add a few milliliters, about 20 to 30 milliliters of the zinc sulfate solution and copper sulfate solution into your beaker. You will grab the copper electrode and you will put it into the zinc sulfate solution, and then you will grab the zinc electrode and place it into the copper sulfate solution. Make sure you observe and record your observations in your lab manual as you begin the experiment and 5 minutes after. After the 5 minutes have passed, you will go ahead and remove the electrodes and dip them into the nitric acid solution to clean them. We will reuse them again. And make sure that anything that you put into the nitric acid solution is well rinsed with deionized water. Okay. Now, moving to part C of the experiment, you will go ahead and discard the copper sulfate solution in the appropriate waste container. All right, and after rinsing the electrodes, you're gonna save the zinc sulfate solution. Actually, for this part, I'm gonna need 12 milliliters, so let me go ahead and pour it now. For this part of the experiment, you will need to know how to use the voltmeter. Make sure you turn the switch to 250 ACV. The red wire will go to the center jack and the black wire will go to the bottom jack. Okay. After this, you can go ahead and turn it on. The red wire will go to the cathode part of the electrochemical cell, and the black wire will be going to the anode. And we can go ahead and begin with our experiment. You had the zinc sulfate solution, and now you have your porous cup. You will go ahead and add 12 milliliters of the copper sulfate solution. All right. And then you're gonna add your electrodes. Okay, so the copper electrode will go to the copper sulfate solution. The zinc electrode will go to the zinc sulfate solution. Make sure that you have your voltmeter or multimeter ready to go, because you need to make the measurement as soon as you put the cup into the beaker. All right, so make sure you're ready to start. So after taking your measurement, you can go ahead and put the voltmeter away so it doesn't get wet. You will go ahead and rinse your electrodes once again, putting them into the nitric acid solution. Always rinse them with deionized water at the end, and you'll be saving them for later. The copper sulfate solution, go ahead and save it for later. You will be using it further down this experiment. Again, you will clean your porous cup with the nitric acid. And rinse it with DA water, always. The same experiment will be repeated with iron sulfate solution and magnesium sulfate solution. Make sure that in between each experiment, before you change the solutions, rinse properly with nitric acid and DA water, as well as the electrodes. After cleaning your porous cup, make sure you have 2 copper electrodes. They have been cleaned in nitric acid solution and rinsed with DA water. You should have about 10 to 12 milliliters of the copper sulfate solution. Pour it into the porous cup. In a beaker, you should have about 20 milliliters of the copper sulfate solution, and you will add about 2 milliliters of 1 molar sodium hydroxide solution. This will form a precipitate. Make sure you mix it and allow the solid to settle down. Once that has happened, you will grab both copper electrodes, place them into the copper sulfate solution, have your voltmeter or multimeter ready to start taking the measurements right away. And observe the measurements and record them into your lab manual. After you have done this, go ahead and turn off your multimeter. You will grab the electrodes, dip them in nitric acid, and rinse them with water. Now that you are done with your experiment, you can go ahead and dispose of everything. Make sure that all your electrodes are cleaned in nitric acid solution and rinsed with DA water. The copper sulfate solution should go into the appropriate waste container. Your TA will let you know. And the nitric acid solution should go into the acid waste container previously labeled.